Next topic, homeostasis. Homeostasis is a fundamental process by which organisms maintain a stable internal environment despite changes in the external environment. It involves a complex set of interactions between the nervous system, endocrine system, and other organ systems in the body. The term homeostasis was coined by Walter Bradford Cannon in 1930, who described it as the maintenance of steady states in the body. There are several key concepts and mechanisms involved in homeostasis, like feedback loops. Homeostasis is regulated by feedback loops, which involve sensors that detect changes in the internal environment, control centers that receive information from the senses and determine the appropriate response, and effectors that carry out the response. Set points. Homeostasis is maintained around a set point, which is a specific value or range of values for a particular physiological parameter, such as body temperature, body glucose level, or pH. Then negative feedback. Most homeostatic mechanisms involve negative feedback, in which the response opposes the initial stimulus. For example, if body temperature increases, the body responds by increasing sweat production and dilating blood vessels to release heat and cool the body. Then positive feedback. In some cases, homeostasis may involve positive feedback, in which the response reinforces the initial stimulus. For example, during childbirth, the release of oxytocin from the pituitary gland stimulates uterine contractions, which in turn stimulate further oxytocin release and stronger contractions. Then multiple organ systems. Homeostasis involves interactions between multiple organ systems, including the nervous system, endocrine system, respiratory system, cardiovascular system, and renal system. Examples of homeostasis includes the regulation of body temperature, blood glucose level, blood pressure, and pH balance. Imbalances in homeostasis can lead to a wide range of physiological disorders such as hypothermia, hyperglycemia, hypertension, and acidosis. Importance of homeostasis. Homeostasis is essential for the survival and proper functioning of organisms. It allows organisms to maintain stable internal environments despite changes in the external environment and to respond appropriately to changing conditions. Homeostasis also helps to ensure that physiological processes are efficient and effective, and that cells and tissues receive the nutrients and oxygen they need to function properly. In summary, homeostasis is a complex and essential process by which organisms maintain a stable internal environment and involves a variety of feedback loops, set points, and interactions between multiple organ systems. Its importance lies in the fact that it enables organisms to survive and function properly in a constant changing environment. Next, taxonomy. Animal taxonomy is the science of naming, describing, and classifying animals. It is important in understanding the diversity of the animal kingdom and studying their evolution, genetics, behavior, and ecological roles. Animal taxonomy is based on the principles of evolutionary biology and uses a hierarchical system of classification that groups animals based on their shared characteristics. The main levels of animal taxonomy are kingdom. Animals are classified into the kingdom Animalia, which includes all multicellular animals. Then phylum. The kingdom Animalia is further divided into phyla based on certain anatomical and developmental features. For example, chordates are a phylum of animals that have a notochord, a dorsal hollow nerve cord, and gill slits at some stage of their development. Then class. Each phylum is further divided into classes based on characteristics such as body plan, presence or absence of certain features, and developmental patterns. For example, the class Mammalia includes animals that have hair or fur, mammary glands, and a four-chambered heart. Then order. Classes are divided into orders, which are groups of animals that share certain characteristics. For example, the order Carnivora includes animals that are meat eaters and have specialized teeth and claws. Then family. Orders are divided into families, which are groups of animals that share more specific characteristics. For example, the family Felidae includes cats and is characterized by retractable claws, a short snout, and keen senses. Then genus. Families are further divided into genera, which are groups of animals that are closely related and share many characteristics. For example, the genus Panthera includes big cats such as lions, tigers, and leopards. Then species. Finally, each genus is divided into one or more species, which are groups of animals that share the same basic structure, behavior, and genetic makeup. For example, the species Panthera leo includes all lions. Then animal taxonomy is important for many reasons, such as conservation purpose. By understanding the relationship between different animal species and their evolutionary history, we can better understand the impact of environmental changes on their survival and make steps to protect them. Then agriculture. Understanding the taxonomy of livestock and crops is important for breeding programs and disease control. Then biomedical research. Animal taxonomy is important for studying the genetics, physiology, and behavior of animals and for developing new treatments for diseases. Then education. Animal taxonomy is an important part of biology education as it provides a framework for understanding the diversity of life on Earth. Then plant taxonomy. Plant taxonomy is the science of describing, naming, and classifying plants. It is an important branch of botany that helps in identifying and understanding the diversity of plant life on Earth. Taxonomy provides a systematic framework for organizing and studying plants based on their evolutionary relationship, morphological characteristics, and genetic relationships. Here are some of the major points about plant taxonomy, like classification. Plant taxonomy involves the classification of plants into various groups or taxa based on their characteristics. The different taxa are arranged in a hierarchical order, which each level representing a different rank, such as species, genus, family, order, class, division, and kingdom. Then nomenclature. Plant taxonomy also involves the assignment of scientific names to plants based on their characteristics. The scientific names are used worldwide to avoid confusion caused by common names that may differ from one language to another. Then identification. Taxonomy helps in the identification of plants based on their characteristics. The morphological features such as leaves, flowers, fruits, and stems are used for plant identification. Then phylogenetics. Taxonomy also deals with the evolutionary relationships among different plant groups. Phylogenetics uses molecular and genetic techniques to study the genetic information among different plant taxa. Then importance. Taxonomy is important for the conservation and management of plant biodiversity. It helps in identifying and documenting the diversity of plant life, which is essential for their conservation. It also helps in plant breeding, agriculture, and horticulture. Then examples of plant taxa include species. A species is a group of plants that share common characteristics and can interbreed to produce viable offspring. A genus is a group of closely related plant species that share a common ancestor. Then family. A family is a group of genera that share common characteristics and have a common ancestor. Order is a group of closely related plant species, families that share common characteristics. A class is a group of closely related plant orders that share common characteristics. A division is a group of closely related plant classes that share common characteristics. Kingdom is the highest taxonomic rank and includes all plants. Overall, plant taxonomy is crucial for the study and understanding of plant diversity, evolution, and conservation. It provides a systematic framework for organizing and studying plants and is important for many applications, including agriculture, horticulture, and medicine, molecular biology, and cell biology. Replication. Replication is a process of copying a DNA molecule to produce two identical copies of the genetic material. This process is essential for cell division, growth, and the transmission of genetic transformation from one generation to the next. There are several factors that can affect the replication process, including drugs that can interfere with DNA synthesis. The process of replication can be divided into three main stages, like initiation, elongation, and termination. In initiation stage, a protein complex called the origin recognition complex (ORC) binds to the DNA at specific sequences known as origins of replication. This helps to unwind the DNA double helix and recruit other proteins to form a replication complex. Then elongation. During the elongation stage, the replication complex uses the original DNA strand as a template to synthesize a new complementary strand of DNA. This is accomplished through the action of an enzyme called DNA polymerase, which adds nucleotides to the growing strand in a specific order determined by the base pairing rules of DNA. Then termination. The termination stage involves the completion of DNA synthesis and the dissociation of replication complex from the DNA. Then drug. There are several drugs that can interfere with DNA synthesis and inhibit the process of replication. An example of such a drug is hydroxyurea, which works by inhibiting the enzyme ribonucleotide reductase. This enzyme is responsible for converting ribonucleotides to deoxyribonucleotides, which are the building blocks of DNA. By inhibiting this enzyme, Hydroxyurea reduces the amount of deoxyribonucleotides available for DNA synthesis, thereby slowing or stopping replication. Another example of replication inhibiting drug is amphidecolin, which binds to DNA polymerase and prevents it from adding nucleotides to the growing DNA strand. This drug is often used in research studies to investigate the process of replication and the factors that affect it. Next, transcription is the process of copying genetic information stored in DNA into RNA. It is a fundamental process in gene expression and is essential for the synthesis of proteins and other cellular components. The general steps involved in transcription are initiation, elongation, and termination. Initiation is the first step in transcription, where RNA polymerase enzyme binds to promoter region of DNA and unwinds the double helix. The promoter region is the specific sequence of nucleotide that signals the start of a gene. Once the RNA polymerase is bound to the promoter region, it starts synthesizing RNA in the 5' to 3' direction. Then elongation is happening. In this step, RNA polymerase moves along the DNA template strand and synthesizes RNA in the 5' to 3' direction. The RNA molecule is complementary to the DNA template strand, and the base pairing occurs between the RNA nucleotides and the DNA template strand. Then termination. This is the final step in transcription, where RNA polymerase reaches the termination sequence and dissociates from the DNA template strand along with the newly synthesized RNA molecule. Transcription is a highly regulated process and involves the coordination of various transcription factors and regulatory elements. These factors and elements can influence the initiation, elongation, and termination of transcription and can also affect the rate of gene expression. One of the key factors in transcription is RNA polymerase, which is responsible for catalyzing the synthesis of RNA. There are different types of RNA polymerases, and each plays a special role in transcription. RNA polymerase II, for example, is responsible for transcribing protein-coding genes, while RNA polymerase III transcribes non-coding RNAs. Transcription is also an important process in various biological processes, including development, differentiation, and response to environmental stimuli. For example, during development, certain genes are transcribed at specific stages to regulate the formation of tissue and organs. In response to environmental stress, the expression of certain genes can be upregulated or downregulated through transcriptional regulation. Understanding the transcription process is essential for understanding gene expression and its regulation. It has also provided insights into various human diseases, including cancer, where abnormal transcription regulation can lead to the uncontrolled growth of cells. Then ribosome profiling. Ribosome profiling, also known as ribosome footprinting or deep sequencing of ribosome protected mRNA fragments, is a technique used to identify the exact locations of ribosomes on an mRNA molecule. This technique can be used to study the process of translation, the mechanisms of regulation of gene expression, and identification of novel coding and non-coding RNA molecules. The basic principle of ribosome profiling involves treating cells with a translation inhibitor such as cycloheximide, which arrests the ribosome on the mRNA molecule. The cells are then lysed and the ribosome mRNA complexes are isolated and digested with nucleases to generate a pool of ribosome-protected mRNA fragments. These fragments are then converted into cDNA libraries, which are sequenced using high-throughput sequencing technologies. The sequence reads obtained from the sequencing are then mapped to a reference genome or transcriptome to identify the exact locations of ribosomes on the mRNA molecule. Ribosome profiling has several important applications in molecular biology and cell biology. Some of the major important points and examples of its application are identification of novel coding and non-coding RNA molecules. Ribosome profiling can be used to identify novel coding and non-coding RNA molecules that are translated by the ribosome or associated with it, respectively. Then analysis of translational efficiency. Ribosome profiling can be used to measure the translational efficiency of different mRNA molecules under different cellular conditions, such as during different stages of development or under different stress conditions. Then a study of translational regulation. Ribosome profiling can be used to study the mechanisms of regulation of translation, such as by identifying the cis and trans-acting factors that modulate the translation of specific mRNA molecules. Then analysis of alternate splicing. Ribosome profiling can be used to analyze alternative splicing events and their impact on the translation of mRNA molecules. Then identification of drug targets. Ribosome profiling can be used to identify potential drug targets by analyzing the impact of different drugs on the translation of specific mRNA molecules. So in summary, ribosome profiling is a powerful technique that has revolutionized the study of translation and gene expression regulation. Its application is broad, ranging from basic research to drug discovery, and it has the potential to provide valuable insights into many biological processes. Then, translation is the process of synthesizing a protein from an mRNA template, which we have already mentioned. It occurs in the ribosome and involves the participation of various proteins and factors. Then, ribosome profiling is a technique used to study translation at the genome-wide level. It involves isolating ribosome-protected mRNA fragments which represent the sites of active translation and sequencing them to identify the corresponding regions of the genome. Ribosome profiling can provide insight into translational efficiency, then codon usage and ribosome dynamics, among other aspects of translation. It has been used to study translation in a variety of organisms, including bacteria, yeast, plants, and animals. Then mTOR kinase. mTOR, mechanistic target of rapamycin, is a serine threonine kinase that regulates protein synthesis and other cellular processes such as autophagy and metabolism. It is activated in response to growth factors and amino acids and integrates signals from various pathways to promote cell growth and proliferation. mTOR controls translation by phosphorylating the translation machinery, including ribosomal proteins and initiation factors. Then dysregulation of mTOR signaling has been implicated in various diseases including cancer, diabetes, and neurodegenerative disorders. Then translation targeting drugs. Several drugs target translation as a means of inhibiting protein synthesis in cancer cells. These drugs include inhibitors of eukaryotic initiation factor 4E, ELF4E, which binds to the 5' cap of mRNA and recruits the ribosome to initiate translation. Inhibitors of the eukaryotic initiation factor 4A, like ELF4A, which unwinds the mRNA secondary structures to facilitate ribosome scanning, and inhibitors of the ribosome itself, such as cycloheximide and puromycin, by inhibiting translation. These drugs can suppress tumor growth and proliferation. However, they can also have off-target effects on normal cells and cause toxicity, highlighting the need for selective targeting strategies. Then, operon. The operon is a genetic regulatory system found in prokaryotic cells. It consists of a cluster of genes that work together to perform a specific function along with the promoter region and the operator region. The operon controls the expression of these genes by turning them on or off in response to environmental conditions. The promoter region is a DNA sequence where RNA polymerase binds to initiate transcription. The operator region is located between the promoter and the genes and contains a DNA sequence that is recognized by a regulatory protein known as a repressor. The repressor protein binds to the operator region and prevents RNA polymerase from transcribing the genes. There are two types of operons: inducible operons and repressible operons. Inducible operons are normally turned off, but can be induced to turn on when specific molecules known as inducers bind to the repressor protein and inactivate it. The lac operon, which controls the metabolism of lactose in E. coli, is an example of an inducible operon. Then repressible operons are normally turned on, but can be repressed and turned off when specific molecules known as co-repressors bind to the repressor protein and activate it. The TRP operon, which controls the biosynthesis of amino acid tryptophan in E. coli, is an example of a repressible operon. The operon concept is important in understanding the regulation of gene expression in prokaryotic cells. By controlling the expression of groups of genes together, the cell can coordinate the production of protein needed for specific functions. This allows for efficient use of cellular resources and enables the cell to respond to changes in its environment. Then the study of operons has also provided insights into the mechanisms of gene regulation in eukaryotic cells. While eukaryotic cells do not have operons, they use similar regulatory mechanisms involving transcription factors and DNA binding proteins to control gene expression. Then phage regulation. Phages are viruses that infect bacteria, and their replication cycle involves a lytic or lysogenic cycle. The phage replication cycle can be regulated by various mechanisms that enable the phage to modulate its own gene expression in response to environmental cues. So, lysogeny, some phages can integrate their genome into the bacterial chromosome and establish a stable dormant state called lysogeny. This is achieved by a phage-encoded repressor protein that binds to specific operator sequences and prevents the expression of lytic genes. Under certain conditions, such as DNA damage or nutrient depletion, the repressor can be inactivated or degraded, leading to the activation of lytic genes and the onset of a productive infection. Then positive regulation. Phages can also use positive regulatory mechanisms to activate the expression of specific genes, such as those involved in DNA replication or capsid assembly. This is often accomplished by phage-encoded transcriptional activator proteins that bind to specific promoter sequences and recruit the host RNA polymerase to initiate transcription. Then anti-repressin. Some phages can overcome the host repression machinery by producing anti-repressor proteins that bind to the repressor and disrupt its ability to bind DNA. This allows the repressor of lytic genes and the induction of a productive infection. Then lysogeny decision. Some phages can use a genetic switch to determine whether to enter the lytic or lysogenic pathway. This switch is often controlled by a phage-encoded protein that can either activate or repress the expression of genes involved in DNA replication and integration. The decision to enter lysogeny is often influenced by the availability of host resources and the presence of competing phages. Then coevolution with hosts. Phages and bacteria have been co-evolved for billions of years, and this has led to the evolution of various mechanisms that enable phages to regulate their replication cycle and maximize their fitness. For example, some phages can use specialized proteins to degrade the most restricted modification systems that are used to defend against foreign DNA. Overall, the regulation of phage replication is a complex and dynamic process that involves various mechanisms to ensure the efficient use of host resources and survival of the phage populations. Then solute transport. Solute transport refers to the movement of ions and molecules across cellular membranes. This process is essential for many physiological processes in both eukaryotic and prokaryotic organisms, such as nutrient uptake, waste removal, and signal transduction. There are several types of solute transport mechanisms, including passive diffusion, facilitated diffusion, primary active transport, and secondary active transport, etc. Then there are passive diffusion. In this process, solutes move from an area of high concentration to an area of low concentration, following the concentration gradient without requiring energy input. Examples of solutes that move passively are oxygen and carbon dioxide in the process of gas exchange in plants and animals. Then facilitated diffusion. This process is similar to passive diffusion, except it involves the assistance of membrane-bound proteins known as transporters or carriers to move solutes across the membrane. The transporters provide a pathway for solutes to pass through the membrane and are often specific to a particular type of solute, such as glucose or amino acids. Then primary active transport. This process involves the use of ATP to move solutes against the concentration gradient from an area of low concentration to an area of high concentration. This process is facilitated by specific membrane proteins called pumps, which use ATP hydrolysis to create an electrochemical gradient across the membrane. Then secondary active transport. This process is similar to primary active transport, except it uses the electrochemical gradient created by primary active transport to move solutes across the membrane against their concentration gradient. Then secondary active transport can be either symport, in which two solutes move on the same direction, or antiport, in which two solutes move in opposite directions. Examples of solute transport processes in animals include the uptake of glucose by intestinal cells through facilitated diffusion, the movement of sodium and potassium ions across nerve cell membrane through primary active transport, and the reabsorption of glucose and amino acids in the kidney through secondary active transport. In plant, solute transport processes are essential for the uptake of water and nutrients from the soil, as well as the transport of sugars and other metabolites throughout the plant. Examples include the transport of ions across root cell membranes through primary active transport, and the movement of sucrose from leaves to roots through phloem tissue via active transport. Then solute transport refers to the movement of dissolved substances such as ions or molecules across a cell membrane. This process is essential for the survival of cells and organisms, as it allows them to acquire nutrients, eliminate wastes, and maintain homeostasis. These topics we have learnt earlier. Then protein targeting. Protein targeting is a process by which newly synthesized proteins are transported to their designated destinations within a cell. This process is crucial for the proper functioning of a cell, as proteins play essential roles in a wide range of cellular processes. Then major points: proteins are synthesized on ribosomes and transported to various organelles or compartments within the cell. Then protein targeting can occur co-translationally and post-translationally. Proteins are targeting signals that direct them to their correct destination. There are several pathways for protein targeting, including the secretory pathway, mitochondrial targeting pathway, chloroplast targeting pathway, and peroxisomal targeting pathway. The signal recognition particle SRP pathway targets proteins to the endoplasmic reticulum. The TOM and TIM complexes are involved in targeting proteins to the mitochondria. The TAT pathway targets proteins to thylakoid membrane and chloroplast. The PTS1 and PTS2 pathways target proteins to peroxisomes. Then protein targeting is essential for the proper functioning of a cell. Mislocation of proteins can lead to a wide range of diseases, including Alzheimer's disease, cystic fibrosis, and diabetes. Understanding the mechanisms of protein targeting is also very important. Then cell cycle. Cell cycle is the process by which a cell grows, replicates its DNA, and divides into two daughter cells. This process is controlled by a complex network of regulatory proteins and checkpoints that ensure the proper progression of the cell cycle. Major points include the cell cycle consists of four main stages like G1, GAPE1, S synthesis phase, G2 or GAPE2, and mitosis phase. So that's all about this topic. Let's learn other topics tomorrow. Thank you for listening.